I'm Daniel Levine, and this is the Bio Report.
Cancer immunotherapies have shown great promise, but many tumors can develop resistance as the changing tumor microenvironment can help cancers evade detection and allow cancer cells to proliferate. Teon Therapeutics is developing immuno-oncology therapies for difficult-to-treat cancers that are designed to have the dual effect of restoring immune activity while suppressing cancer cell proliferation. We spoke to Serge Messerlian, CEO of Teon Therapeutics, about what happens when immunotherapies fail, the unique approach Teon is taking, and how the dual activity of its cancer therapies can restore immune activity while suppressing cancer cell proliferation. Serge, thanks for joining us.
Absolutely. Thanks for making the time, Danny.
We're going to talk about Teon Therapeutics, its immuno-oncology therapies and development, and its unique approach to address difficult-to-treat cancers. Before we get to what the company is doing, though, I wanted to start with you. You joined about a month ago from Janssen Oncology, which I imagine is a much better resource company. What attracted you to Teon? What was the opportunity you saw?
Oh, thanks, Danny. Really a few things, to tell you the truth. First and foremost, it really started with the quality of the team. As I was having conversations with the organization, the R&D team is extraordinary. They have very extensive experience with specific GPCR molecules, G protein coupled receptors. They spent their entire careers working with these types of receptors, in particular, the chemistry and the modulation of those receptors. And so I think depth of expertise, counts for a lot. And I think it also says a lot in that, frankly, 3.5, 4 years ago, there was no company. And to go from nothing to, a company where we have one molecule in the clinic now and soon a second, I think really says a lot about the insights that the R&D team has and the level of their judgment in terms of designing and developing molecules that hopefully are effective. But that I think was a very important point. And then, insofar as the quality of the science goes, I really saw, the idea of targeting G protein coupled receptors is really an untapped space in oncology. It's well known in many other diseases, as drug targets, but for oncology, it's lesser known. And I think with the targets that we are looking at in the adenosine space, as well as other spaces, I just think there's a lot of opportunity there to be able to make an impact with the science that the team is presently doing, with the drugs that we're presently developing. So I think taken together, it gave me a lot of confidence that this was the right move and that Teon was the right company with the right aspiration ahead.
Immunotherapies have been an exciting emerging area of cancer, but they're only effective in a small group of patients. What happens in the tumor microenvironment that allows tumors to grow resistant to immunotherapies?
Well, that's a pretty complex question, and I can think of, a number of ways in which resistance, either primary resistance or secondary resistance, acquired after kind of being on an immunotherapy, take place. But it's a very complex field. But just to try to break it down, in some ways, there are either kind of reasons that do not allow a T cell to become reactive, right? Either tumors don't present what's called neoantigens, which sort of demonstrate to the body that they are foreign, or that how those antigens get presented to the immune system. There may be sort of deficiencies there. There could be deficiencies in terms of kind of stimulatory signals that are to sort of amp up the immune system. They can become impaired. The tumor microenvironment, kind of secondly, is a pretty hostile place, for the immune system. Various cells can be drawn into that tumor microenvironment to inhibit immune response, like MDSCs, which are suppressor cells, myeloid-derived suppressor cells, and other such cells. So really, there's a host of mechanisms that create, ultimately resistance. And, you're right. Immune therapies broadly hold a lot of promise, but they may not work for everybody, and they may not be durable, importantly enough. And that has to do with, perhaps ways that tumors are able to evade or sort of limit T cells to become memory cells to have a durable response. But, taken together, it's naturally a very complex space. We haven't solved everything in the space, and that means that there's a lot of opportunity ahead, certainly to develop more therapies that can be even more helpful for patients ultimately.
Teon is developing therapies that both target metabolic signaling pathways to restore anti-tumor immunity and also suppress cancer cell proliferation. You're actually using small molecule therapies to do this by targeting, as you mentioned, the G protein couple receptor. This is a target that's been exploited to treat a large number of conditions, but generally not cancers. Why is that?
I think maybe for a few reasons, that I can think about at least. Generally, as we think about the evolution of cancer therapy, it wasn't really that long ago that cancer therapy development has taken the turn to become more sophisticated as we sort of entered the realm of developing targeted therapies, as we entered the realm of immunotherapies and the like. I mean, these are things that have kind of largely transpired, let's say, over the course of the past decade or so. Now, having said that, I think when you look at kind of G protein coupled receptors, there's I think 800 plus genes that are ascribed to that. A good number of them are sort of well understood and are associated with other diseases, but insofar as oncology goes, they're not quite as well understood. And moreover, in order to really understand them, it's not just kind of the receptors that you need to understand. Importantly, you need to understand the ligands or the molecules that sort of connect with these receptors to turn them on or turn them off, depends on kind of what's happening. But all this to say, the ligand and the receptor, they're kind of like a lock and key, and they need to work together. And one needs to understand both sides of that equation. One needs to understand how is the lock working, and what does it do, and how does it function, and then what does the key do, and how does it work, and how is it designed, and how does it interact with the lock, essentially. And a lot of that isn't yet well understood, or that pieces are missing. There are many G protein coupled receptors to which the ligand is unknown. And so it's really difficult to understand kind of its function within a tumor conceivably, and what it does and how it works. And then more importantly, what to do about it to either, sort of block it or inhibit it. So kind of all this to say there's still a lot to be learned in the realm of oncology as it relates to G protein coupled receptors.
What's the Warburg effect, and what's the significance of this with tumor microenvironment?
Yeah. So the Warburg effect is, it's something that's been known for a long time. Maybe 100 years. I don't know the exact time by which that theory was developed. But in fact, what it really is about is that tumors have the ability to create their own energy through glycosylates, and that is a little bit different than what normal cells do. Now, normal cells can also function in an anaerobic state, through sort of glycolytic enzymes, right? But glycosylates is it's an inefficient way to produce energy. Most normal cells produce their energy through, you likely well know the Krebs cycle or the tricarboxylic acid cycle, et cetera, which is a much more effective way. It's effectively called oxidative phosphorylation. But tumors have adapted in some way, whether it's in an anaerobic state or aerobic state, to predominantly draw their energy through glycolysis. So that's effectively what the Warburg effect is. And what it means, kind of relative to tumor microenvironment, is because they're able to sort of digest glucose in the cytosol of the cells, they're able to sort of they create as a byproduct a lot of lactate. And lactate is able to create, sort of acidity and kind of help create, part of this toxic or hostile environment that I was telling you about as part of the TME or the tumor microenvironment. So, there are different strategies presently being employed by different organizations to see, is there a way to inhibit or limit that glycolytic sort of process in the tumor, and then halt the tumor progression as a result. But it's one strategy amongst many that organizations like us are looking to exploit, which is metabolism, broadly of a tumor.
Well, you're trying to antagonize metabolic pathways in both tumor cells and immune cells. What are you actually doing? Are you targeting specific enzymes?
Yeah, so one of our programs, our lead program, which we call TT702, is an adenosine 2B, 2 beta receptor antagonist. So what is that? There is a very important metabolic pathway, which is the adenosine pathway. And what we are trying to do is that adenosine that gets produced, ultimately, by the tumor ends up kind of collecting in the tumor microenvironment, and then connecting to adenosine 2B receptors in immune cells. And what it does is it inhibits the immune system, effectively shuts it down. And our job is to be able to create antagonists that are highly selective to 2B and highly potent, such that we're able to block that effect. And if we're able to block that effect, then our hope and expectation is that it'll turn the immune system back on, and prime T cells and other cells conceivably to start to act against the tumor. And that's really what our job is, whether it's TT702 or frankly any of the other sort of targeted portfolio molecules that we're developing. They're all kind of geared to do the same type of thing, which is inhibit tumor metabolism in some way, and then kind of reverse the suppression of the T cells or the immune system.
Given the mechanism of action, do you expect to use these as monotherapies, or do you see them being better used in combination with other immunotherapies or other therapies?
Yeah, well, you well know, I think in many respects, oncology is a combination game. Now, having said that, our plans are to demonstrate effect in all of our molecules as monotherapies. Our second program, not TT702, that's the lead program, but the second program that we're working on, something called TT816, is actually a small molecule immune checkpoint inhibitor. And it's something that we're very excited about. Now, we haven't released what the target is of this molecule, but as an example, our plan is to demonstrate monotherapy effect with that small molecule immune checkpoint, and to study it in combination with other types of immune checkpoint inhibitors like a PD-1, PD-L1. So, our thinking is that really to get the most utility ultimately for patients, that we need to study it in combination as well as monotherapy.
Existing immunotherapies have been more successful in liquid than solid tumors. Do you expect your approach to be agnostic to that?
Well, we don't know yet. So far, I will say, as I think about the immune, checkpoint inhibitors that are out there, they've done very well for patients in solid tumors. So our thinking is, really to start in the solid tumor space and to build on that kind of success on the science that is understood and known in the solid tumor space. But certainly as we evaluate our scientific programs and priorities, definitely myself, having spent time in the hematology realm as an example, have interest in exploring in hematology. So, but we can't boil the ocean. We got to start somewhere and learn. And we've chosen solid tumors as a starting point, but naturally open to considering any and all avenues to which patients could benefit from, from our scientific exploration.
You talked a bit about TT702. This is your lead experimental therapy. You're conducting a phase 1/2 clinical trial on a range of difficult to treat cancers. How does this study work, and how will it define the development path forward?
Well, really what we're looking to do is to evaluate, certainly in the phase 1, first off, the safety and the appropriate dosing of the molecule. And that's really what the goal of the phase 1 is. And it's a phase 1 that's sort of defined to assess the molecule in mixed tumor types. And I think as we learn, how this therapy may perform, through the phase 1 in humans, to be able to determine a safe dose, I think that's really the kind of the most important goal that we can achieve. Thereafter, I think that it'll really depend on kind of what patients we want to select to expand into cohorts, into phase 2 and other types of expansions. But for us now, what's really important is that we get a good feel for the safety and the tolerability of the program to ensure that any further science that we do in people is done in a very safe and controlled manner.
You're doing this study in collaboration with Cancer Research UK. Are they funding the study, and how far might they take this in development with you?
Yes, they are. They are supporting the funding of the study, and presently our scope with them is to do this in partnership for the phase 1 and the phase 2. So that's kind of the scope that we have presently.
What's known about the safety and efficacy of the therapy at this point?
Can't really say yet. We're still in the early goings of it, and we're looking to advance the phase 1 over the course of the year to have sufficient data to say something about the safety aspect of the program. So you'll have to invite me back at a later time, and I'm happy to share the data as we have it available.
Well, you raised $30 million in a venture round in February 2021. How far will existing funding take you, and what's the plan for raising money from here?
Yeah, we had a very successful raise last year. It really set the organization up for success to be able to achieve these short-term value inflecting sort of milestones that we have, getting into the clinic, filing INDs, et cetera, advancing some of our preclinical pipeline. So the funding will take us, largely to the end of the year, and we have flexibility around that. We have very supportive investors, which we're really, really lucky to have in this challenging funding environment. And at some point, certainly we'll be considering another raise, and we're in the process of evaluating what are our options in terms of doing that. So, more to come on that.
Serge Messerlian, CEO of Teon Therapeutics. Serge, thanks so much for your time today.
Thank you, Danny. It was a real pleasure to speak to you, and I really enjoyed the conversation.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.